The kidneys are bean-shaped organs located on the either side of the vertebral column in the upper posterior part of the abdominal cavity. They are positioned between the 12th thoracic and 3rd lumbar vertebrae, with the right kidney slightly lower than the left due to the position of the liver. Now I want you to locate peritoneal cavity. Yep, right there. And this peritoneal cavity, also known as abdominal cavity, is lined with the peritoneal membrane. Can you locate parietal peritoneum, that is outer layer, and the visceral peritoneum, that is inner layer? So together they are known as peritoneal membrane, which lines the peritoneal cavity of the abdominal cavity. Now, if you look at the location of the kidneys, both kidneys, right and left, they are located behind the peritoneal membrane. And since they are located behind the peritoneal membrane, that's why we say the kidneys are located retroperitoneally. Our kidneys are surrounded by 3 protective layers. I want you to locate renal capsule. Yes, there you go. It is a tough fibrous covering. Now I want you to locate perirenal fat. Yes, there you go. So this perirenal fat is a fatty layer that cushions the kidneys. Next, I want you to locate renal fascia. Correct? It is a connective tissue layer that anchors the kidneys in place. Let us now discuss the composition of our excretory system. So it consists of the main organs, that is kidneys, and its primary function is to filter the waste from the blood, excess water and toxins from the blood, ultimately resulting in the formation of urine. It consists of ureters, which are 2 thin tubes that will transport urine from the kidneys to the urinary bladder. And urinary bladder is a muscular sac that stores urine temporarily. And then it consists of urethra, a tube that carries urine from the bladder out of the body during micturition or urination. The kidney is made up of different layers and parts. The outermost layer, which is labeled here as renal cortex, is the outermost layer, and the filtration of blood occurs in this layer. The parts of nephron that falls in this layer are Bowman's capsule, glomerulus, PCT, and distal convoluted tubule. So this tells us that by any chance, if the renal cortex gets damaged, it can severely affect the functioning of these components. Next is the middle layer, which is labeled as renal medulla. So this right here is the renal medulla, consisting of renal pyramids. Let us see the parts of nephron that falls in this renal pyramids. So the parts of nephron which falls in the renal medulla are loop of Henle and collecting ducts. These renal pyramids contain structures that helps in concentrating the urine, and in between them is known as renal column, and the tip of each renal pyramid is known as renal papilla. From this tip, the urine is released from the collecting ducts into the minor calyx. And all these minor calyces, the funnel-shaped structures, they will collect urine from the renal papillae, which is a plural form, and then they will release it into the major calyces. And all these major calyces combined will form renal pelvis. The renal pelvis then collects urine from the major calyces and directs it into the ureter. And from the ureters, the urine is carried to the urinary bladder. And urinary bladder stores urine. Urinary bladder is composed of detrusor muscle which contain stretch receptors. And these stretch receptors send signals to the brain when the bladder gets full, triggering the urge to urinate. And during urination, or we can also call it as micturition. So during micturition, the internal and external urethral sphincters relaxes, allowing urine to pass through the urethra and get excreted from the system. Let us now shift our focus to nephron. Each kidney consists of millions of nephrons, and without these structured nephron, kidney won't be able to carry out their primary and other functions. That is why nephron are said to be the functional unit of the kidney. And in this discussion, you are going to primarily focus on how every part of nephron are structurally adapted to carry out their functions. The renal artery will branch into smaller blood vessels called arteriole, and the efferent arteriole will supply blood to the glomerulus that needs to be filtered, and after filtering, the blood will be taken away by the efferent arteriole, and efferent arteriole will further divide into blood capillaries that will surround the PCT and DCT, which is labeled as peritubular capillary networks, and further branching occurs. And these network of capillaries that runs along the loop of Henle is known as vasa recta. And in these capillaries, the further exchange of substances, that is reabsorption and secretion, will occur. If you look at the size of the efferent arteriole and efferent arteriole, their size differs. Efferent arteriole is wider in diameter compared to efferent arteriole, which is narrower in diameter. Now, why do you think efferent arteriole has to be wider in size? It is because this wide diameter will allow a greater volume of blood to enter the glomerulus, and this will increase the blood inflow and generate high glomerular hydrostatic pressure within the glomerular capillaries. And this high pressure is required to force the excess substances, toxins, water from the glomerular capillaries into the Bowman's capsule. Smaller diameter of the efferent arteriole will maintain the high glomerular pressure by creating high resistance to the blood outflow from the glomerulus. So this resistance will enhance filtration by preventing rapid drop in pressure, and this will allow more solutes to pass through the glomerular filtration barrier, resulting in the formation of glomerular filtrate. So based on this concept, you can try to answer this question. Differentiate between efferent and efferent arterioles of nephron based on parameters given in the table. So you have to differentiate efferent and efferent arteriole based on their structure and based on their functions. Do try to answer this question as well. The sizes of efferent and efferent arterioles differ from each other. How is this significant to the process of ultrafiltration? And this question as well, what would happen to the volume and concentration of glomerular filtrate if the lumen of the efferent arteriole is wider than that of the efferent arteriole? You can leave your answers or even doubts in the comment section. And in the next video, we will discuss on every part of nephron and how they are structurally adapted to carry out their functions. And if you haven't subscribed yet, do hit the bell icon to get the notification. Until then, take care. See you in the next video.